In this video, I will present the role of lysosomes in Parkinson's disease. Now, Parkinson's disease is a disorder of the central nervous system. The symptoms often include tremors, stiff muscles, memory issues, and even dementia. A major cause of Parkinson's disease is impairment of the autophagy lysosomal pathway. This pathway is responsible for the degradation of aggregated proteins, and so when it is disturbed, there is an accumulation of a protein called alpha-synuclein. We can see that certain regions of human alpha-synuclein have the alpha helical secondary structure. The alpha-synuclein aggregates, which constitute Lewy bodies, form elongated, fibrillar, beta sheet structures that are held together by hydrogen bonds. It is believed that the cell-to-cell transmission of the misfolded alpha-synuclein protein causes the onset and progression of Parkinson's disease. However, the process by which this occurs is poorly understood. A better understanding of the transmission process may allow for therapeutic techniques that inhibit the spread of pathology. In this video, I will begin with background information to familiarize you with the context of major findings. And then, I will present quantitative results suggesting that lysosomal integrity is a rate-determining step in the development of pathology. Now, this is simply a fluorescent image of the alpha-synuclein aggregate constituting a Lewy body. When alpha-synuclein preformed fibrils, also known as PFFs, are introduced to neurons from a brain region called the hippocampus, there is formation of pathological insoluble aggregates through the recruitment of alpha-synuclein from within the neuron. The PFFs can be thought of as infectious seeds in the formation of the alpha-synuclein aggregates. As the misfolded protein seeds recruit more and more alpha-synuclein from within the neuron over the course of days to weeks, a positive feedback loop is established. Through fragmentation from the main beta sheet-rich Lewy body, more infectious seeds are released, which in turn further progresses the development of pathology. The infectious seeds can be released into the extracellular space, where they are trafficked to a recipient neuron through various possible mechanisms, such as nanotubes, exosomes, or simply as misfolded seeds alone, as the diagram shows. The internalized seeds in the recipient neuron are first trafficked to lysosomes. The lysosomes are where the majority of initial seeding of alpha-synuclein from within the neuron is thought to begin before the aggregates break out into the cytosol. Now, there is some controversy over the location of initial seeding events. For instance, one paper that used DStorm, which is an imaging technique that creates super resolution images, reported that no aggregation was seen in the lysosome. On the other hand, a more recent paper overcame a limitation in model systems to ultimately support the hypothesis that the majority of initial seeding events occur in lysosomes. The previous limitation was that extracellular and intracellular fluorescently labeled PFFs were difficult to discern. The signal from the extracellular PFFs is not of value in determining the fate of the PFFs, and so the signal was quenched through the use of a membrane-impermeable fluorescence quencher called trypan blue. By observing that the internalized PFFs co-localize with a lysosomal associated protein, researchers were able to conclude that the vast majority of alpha-synuclein is present in lysosome for days. And in the diagram, red represents lysosomal associated protein, while the green represents alpha-synuclein. To further test the role of lysosomes in the development of pathology, a weak base called chloroquine was used. Lysosomal dysfunction occurs after brief exposure of neurons to chloroquine. In the graph, PBS is simply the control, and 81A signal is reflective of the amount of pathology present. We can see that in the first two control conditions, baseline 81A signal was observed. The takeaways are that PFFs result in pathology, as represented by increased relative 81A signal in the last two columns, and that the rate of development of pathology is accelerated through the use of chloroquine, as represented by the last column. Researchers ultimately concluded that lysosomal integrity could be a major rate-determining step in the spread of alpha-synuclein pathology. Now, the limitations in this conclusion is that a major chemical manipulation of neurons was used, namely chloroquine. To see if the results extend to a more physiologically relevant model, a gene knockdown experiment could be performed. If the knockdown of genes critical for lysosomal function leads to similar results observed in the chloroquine experiment, then the conclusion that lysosomal integrity is a major rate-determining step in the development of pathology would be further supported.